I simulated evolution, and the results will surprise you. In this simulation, we have bacteria whose only functional trait is move up. In an environment where selection pressure can be added, this move up trait is randomly mutating across several generations, with selection pressure being introduced as areas of the environment. Areas highlighted green reduce reproduction time. Areas highlighted red reduce lifespan. With this simulation, the goal is to see whether or not the only functional traits move up through random mutation and selection pressure can produce the functional trait move down, which does not yet exist. I've seen other simulations where they randomly add and match new functional traits that are already programmed in, which can then be selected for. They then present that as evolution. However, this does not accurately reflect evolution. Evolution is random mutation of already existing traits plus selection, not random addition of new traits plus selection. You see, the consensus on general evolutionary view is that random mutation of already existing traits plus selection can produce new functional traits. My view is that it can't. Yes, it does produce different traits, but those traits are just derivatives of the already existing traits. My position is based on a mechanistic understanding of how proteins evolve. DNA encodes proteins, and mutations can only modify the proteins and structural scaffolds that already exist. Evolution operates through variation of existing folds via point mutations, insertions, deletions, duplication, and recombination. But it does not invent entirely new, unrelated protein folds from scratch. Because protein folding is constrained by physics, sequence, structure relationships, and the stability requirements of functional intermediates, a lineage can only evolve new functions that are structurally compatible with the protein folds it already possesses. If a function does not exist even in a rudimentary precursor form within the ancestral proteins, then no amount of random mutation or time can generate that function, because evolution must work through viable intermediates built on existing folds. So my central question to the consensus view is this: do we have any empirical, experimentally supported example where a known ancestral protein evolved through a stepwise mutation-driven pathway into a protein belonging to a completely different, unrelated fold family, with each intermediate remaining functional? As far as I can tell, no such example exists, and this absence of mechanistic evidence suggests that evolution is fundamentally constrained by the precursor folds present in each lineage. For those who hold to the consensus view, who wish to criticize my position, you have to answer the following questions. Number one, can you cite a peer reviewed study showing a known ancestral protein evolving through experimentally tested intermediates into a protein with a different, unrelated fold family. I'm not asking whether new proteins can appear or de novo genes can form. I'm asking whether an existing protein fold has ever been shown to mutate into a different fold family. Number 2. Can mutations produce proteins whose function is entirely unrelated to the function encoded in the ancestral protein's fold without going through intermediates that already possess precursor functions? Number 3. Do you know of any case where protein fold class A has been empirically connected to fold class B through a continuous series of mutational states, each one folding stably? Number 4. Since orphan genes have no Number 4. Since orphan genes have no detectable ancestors, how can they be used as evidence of a fold transition when the actual evolutionary pathway is unknown? Number 5. Experiments with random sequences show that some sequences can fold, but these are not mutation descendants of existing proteins. How do such experiments demonstrate fold evolution from an ancestral protein? Number 6. If a mechanism has never been observed, reconstructed, or experimentally demonstrated, is it scientifically justified to infer that it occurred solely on the basis of deep time? Number 7. If a lineage lacks the structural precursor for a function, such as a specific fold architecture, by what evolutionary mechanisms could it evolve that function?